From Microbe TV, this is TWiEVO, This Week in Evolution, episode 11, recorded on August 30th, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,500 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month. And for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. This episode is also sponsored by Drobo, a family of safe, expandable, yet simple-to-use storage arrays. Drobos are designed to protect your important data forever. Visit drobo.com to learn more. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels Elde.
Good afternoon, Vincent. Great to be here from Elde Lab Studios, as usual.
Do you have beautiful weather out there like we do?
My goodness. Yeah, this is the time of the year. So we're hovering right at around 90 degrees. I guess that's about 32 centigrade. And the great thing about Salt Lake this time of year is the evenings just cool off beautifully, and you just want to sit outside for hours.
Nice. I'm sure you do that and don't get any work done, right?
Yeah, that happens from time to time.
Here it's 29°C, 48% humidity, just a few puffy clouds, no rain in sight. It's pretty nice end of summer here.
That sounds great.
So we have a guest today from the West Coast, from Department of Molecular and Cell Biology, the Howard Hughes Medical Institute at the University of California, Berkeley. Nicole King, welcome to TWiEVO.
Thank you. It's great to be here.
So I understand you know Nels, is that right?
That's right. We've run into each other at a few meetings, and I've followed his research with great interest, and
and vice versa. So yeah, one of these meetings we went to was a recent evolutionary cell biology meeting at Genelia Farms last year, which is a really, really fun one. And I have also been following Nicole's work for many years, and I'm a big fan. So it's really exciting to have her on today. Nice.
I know a couple of people out there. You're at Berkeley. Are you familiar with? I'm blanking. Gosh, I'm just such an old guy. I can't remember names anymore.
That's all right.
Well, they're virologists. You probably wouldn't know them anyway, right?
Oh, no. We have some fun Britt Glansinger here.
Yeah, Britt, of course.
Yeah, fantastic.
And the flavivirus lady, Eva Harris.
Great. Yep. And I'm excited because pretty soon we have Julie Pfeiffer visiting from UT Southwestern. So I'm learning all my virology, hopefully.
So I did a podcast there a couple of years ago as part of one of those student-run meetings. I forgot what they're called.
The Ashland meetings, maybe? I don't know. I don't
remember, but the students invited me, and I did a podcast with Britt and Eva in front of a nice little audience there. So that was fun.
Fantastic.
So today we're going to talk about a paper of yours published in PNAS this past July. Bacterial lipids activate, synergize, and inhibit a developmental switch in choanoflagellates. But before we go into that, I want you to tell us just a little bit about your history, starting from where you're from originally.
Ooh, that is a tough question for me. It turns out there's not an easy answer. So I was born in Florida, and at the time, my dad was an undergrad. So when I tell people all the places I've lived, they often ask me whether I have a military background. But no, it's that I got to move every time there was a change. You know, as we academics move a lot, I experienced that as a kid. So I lived in Gainesville, Florida.
Wow.
And then Indiana, and then Reno, Nevada. That was my high school years. And then I started my own academic career, and I went back to Indiana. I was in Cambridge, Mass, Madison, Wisconsin, and now Berkeley is the longest I've ever lived in one place.
So I'm gathering that you went to school in at MIT, maybe? Is that possible?
No, at Harvard.
Harvard.
Yeah.
Well, not bad,
That's right.
And that was undergrad or graduate school?
That was graduate school.
What did you work on there?
That also was complicated. So I started out, initially I was interested in studying the origin of homeobox genes. And it was a project that I had kind of cooked up myself along with a paleontologist at Harvard named Nicole. And it was a really wonderful experience working with him. I learned a ton, but he obviously didn't have a molecular biology lab. So I would go to another lab to do experiments. And I really didn't have a community. Most graduate students have a lab they're working in and a lot of people to bounce ideas off of. But I was kind of a solo operation, and about 3 years in, for a variety of reasons, I had to face the music that it probably wasn't going to work out, that my project didn't seem to be going anywhere quickly, at least. And so I weighed a lot of options, one of which being dropping out. I thought about following a boyfriend, going off to his postdoc, and then ultimately decided that I would try to find another lab. And here I was 3 years in, so I was going to need to be able to get something done quickly. I had heard amazing things about Rich Losick as an advisor, and I was very interested in the research going on in his lab. So I joined his lab. He was kind enough to take me so late in the game. And I studied the genetics of bacterial sporulation.
Did you know Jonathan Dworkin?
I did.
Because he's here in my department
amazing.
now.
Yeah, no, you can give him a big hello. I thought he was great. We overlapped, yeah.
Oh, cool. So then I presume Wisconsin was for a postdoc, is that right?
That's right. So I had this, I had been studying the origin of homeobox genes originally, and I had learned about these strange organisms that I hadn't previously heard about before called the choanoflagellates. And I realized, there was hardly anything known about them. They were the closest living relatives of animals. And I thought, geez, if I ever had the chance, I'd really like to study choanoflagellates and try to use them as a way to investigate animal origins. So I started looking around, and there weren't any labs that were devoted to their study, or that had really started studying them at a molecular level. So I was kind of not sure what to do. And then I met Sean Carroll, and he was very interested, and he was willing to have me join his lab, where I would try for a year or two to study choanos, and if it didn't work out, I would turn to studying fly genetics, which is something I'd done as an undergrad.
So, that's really interesting, Nicole. So I didn't realize that you were already in that transition from grad school to postdoc, already thinking about choanos. So would you, as our listeners or others are making that transition, what would your recommendations be about being interested in sort of emerging model systems or systems that haven't been developed, in the timing, the challenges of that?
Right. I mean, I was, I think, extremely fortunate. When I think about what failed and when I was in graduate school, a lot of it was the timing and the technology. So I think that it was just too hard to dive into a non-model organism when I was a graduate student, or it was too hard as an individual to do it. But between, I guess that would have been 1996 to 1999, sequencing technology improved quite a bit. The prices, while still exorbitant, came down into the plausible range. RNAi was being applied to more and more organisms. And while it didn't end up working for choanos, at least I thought that it might. So that gave me some hope. I think that, the application of antibodies across organisms, I think the field, you were starting to see more and more examples of people taking tools developed in model organisms and applying them to non-model organisms. And I think the field was ripe. So, strangely enough, I think those 3 years spent in the LOSIC lab allowed me to think more about what I would do in choanos given the opportunity, but also sort of gave the field an opportunity for the technology to catch up.
Yeah.
And that's made a huge difference.
Yeah. And so were you already, when you were starting your postdoc, were you already beginning the genome analysis, or did that come later?
That came later. So once I started, the first thing I did was actually, I unpacked my boxes and then I got back on a plane. I flew to the ATCC, what is it called? ATCC.
American Type Culture Collection.
Type Culture Collection, yes, thank you. And they had a director of protostology, Tom Neerad, who knew how to grow choanoflagellates. And so I spent 2 weeks with him. He thawed every strain that they had frozen down in storage there. And I spent a 2-week crash course on choanos. And then brought all of those species back to the lab and started developing tools. And what I realized was that, in fact, the evidence that choanos were the closest relatives of animals was rather thin. So before, here I had embarked on this adventure with choanos, and I realized maybe the whole rationale was really not well supported. And so I actually had to spend some time convincing myself that they are the closest relatives. And that fortunately held up, and there's, I think at this point, no convincing evidence to the contrary.
And actually, maybe I can interrupt you for a second. What do you mean by closest living relative to the animals? What does that actually mean?
Sure. Okay, so I think one thing that I need to remind everybody is that animals are what we call monophyletic. So the animals are a large group representing all of the descendants of a single common ancestor, which was the first animal. And so that group includes everything from ourselves to flies and worms, of course, but also including sponges and cnidaria and a group of organisms that's getting a lot more attention called the ctenophores. So a lot of these squishy organisms that you see in the ocean, these are animals. And they all together, this group shares a common ancestor that probably evolved over 600 million years ago. So choanos, if we think about the tree of life, are the closest relatives of animals. They diverged from the lineage that gave rise to animals, right before animals first evolved. Right before might be 100 million years ago. We don't really know the timing, but so far, we've found no evidence of any organism that's more closely related to animals. So hopefully that makes sense. So these are our cousins.
Yep. So I guess, you said at the end of when you were starting out in Wisconsin, if it didn't work out, you would switch to flies, but I guess it worked out, right?
Yes, it all worked out. So it turned out choanos were closely related to animals.
Yeah.
I spent some time figuring out which species I wanted to study. And then, which is an interesting story relating to this PNAS paper. And then we embarked on initially EST sequencing, which some people they express sequence tags. That was how we gained a foothold. And then based on those results, we could see that choanos were expressing proteins that were otherwise only known from animals. And these included things like cadherins and tyrosine kinases, proteins that allow cells to coordinate their activity within the multicellular animal. We found them in choanos, and that was enough to convince the Joint Genome Institute that this was an organism that should be sequenced. And so that's how we moved ahead with that.
Are there known viruses of choanoflagellates?
I wish. We have been looking, and we've been. I go to meetings regularly with Forrest Rower and Curtis Subtle, two virologists, and we've sent them samples, and so far we haven't found any.
But there must be, right? Yeah, there have to be.
Right.
It's funny. On one of my other podcasts last week, someone asked. Well, long story, but the bottom line is, are there? We assume that every living thing on Earth has a virus, but wouldn't it be interesting if there was something that did not have a virus? And
right.
I mean, you'd have to look for a long time before you made that conclusion, obviously.
I just don't know how you could ever make that conclusion.
Yeah,
yeah. I actually have, it's interesting, I have a bet with one of Nicole's colleagues at Berkeley, Kathy Collins, who's a great tetrahymena molecular biologist. So when Nicole was a postdoc at Wisconsin in Sean Carroll's lab, I was a grad student in Aaron Turkwitz's lab at Chicago, where we were studying cell biology of Tetrahymena thermophila, the model ciliate. And that's another species that doesn't have any known viruses. And so Kathy Collins and I have a bet where she's taking the side that somehow Tetrahymena is the ultimate immune system and are just immune. And so, but my side of the bet is no, they're like everyone else. They have viruses. We just haven't found them yet. And so the bet continues. And it's an interesting, so the way the bet's working is for every year that I or anyone else doesn't find a virus, I have to send Kathy chocolate. And then if and when viruses are discovered, she has to send me a case of pear-infused vodka. So I've got a really good incentive here
to find something.
Pear-infused vodka? Why pear?
Yeah, I don't know, some fancy Berkeley distillery, I guess.
Oh, I see. So
can I just explore this a little more? What about bacterial pathogens? Because we haven't found any in choanos either, and ciliates and choanos eat bacteria.
Yeah, right. Exactly. Yep. Yeah, it's an interesting question. So for Tetrahymena, at least, they can get sort of pseudo-infected, at least with things like Legionella.
Okay.
But it's not super clear that it's a real full-blown infection. So it's a pretty orthogonal route. But that would be something possibly to try with choanos. Would be something like Legionella, which
we have. We have.
Oh, you have. And it didn't work. Didn't take.
Nope.
Is there any evidence of bacterial DNAs in the choano genome from this ingestion? Yes.
There is a paper, I can't remember, it wasn't from our group. Somebody sifted through the genome and they found rampant evidence for horizontal gene transfer. That's complicated because we can't grow choanos in the absence of bacteria. So you have to really convince yourself that it's truly integrated into the choano genome.
Yeah, like the tardigrade story, right?
Yeah.
That was a tough one.
Contamination.
Yeah.
So, Nicole, when you were at Wisconsin, did you overlap with Margaret McFall-Nye?
I didn't. She came later, but I've gotten to know her since then because of all of our interest in host microbe interactions.
Yeah. And now, of course, she's gone again, I understand, right?
I know.
Oh, well.
Now, I'm hoping to visit. I've never been to Hawaii, so. Intent.
Right. So let's talk about the paper. Maybe the first thing you could do is pronounce the two life forms.
The stars?
Yes.
Sure. The stars of the show. So the choanoflagellate is Salpingoica rosetta. And because it's such a bear to pronounce, we just call it S. rosetta.
Okay.
We think if C. elegans can be C. elegans, the virus can be S. rosetta. So that's the choanoflagellate. And the bacterium is called Algoriphagus. That's the genus. And I remind people it sounds like Al Gore phagus. And then the species name is Machiponganensis, which refers to the Native American name for the island where it was found with S. rosetta. So I recommend we call it S. rosetta for the choano and Algoriphagus for the bacterium.
Algoriphagus, yeah. Okay, that's good. Yeah. I'm gonna confuse them anyway, but
okay.
So maybe you can give us the backstory on this pair.
Sure. So if I can back up to my first 2 weeks as a postdoc, I'm out at the ATCC, and what I really want to study are colonial choanoflagellates, ones that can become multicellular. But there were, in fact, none in culture anywhere. And there was one that had been described to be multicellular, and it had lost the ability to become multicellular in the lab. So, that just wasn't going to happen during my postdoc. But while I was at the ATCC, another group was there at the same time, and they were looking at bacterial diversity. But they had all these water samples they were going through, and Tom Nerad, who I was working with, happened to look at their water samples, and he said, "Hey, there's a whole bunch of colonial choanoflagellates in here." And because I was there and the timing was right, he said, "If you want, I can just isolate these for you, and we'll freeze them down, and you can study them." And I was like, "Okay." So, it's just incredible timing. So he froze it down for me, and I saw them. I took pictures of them as multicellular organisms, straight, fresh from nature. And I brought them back to Madison and started up a culture, and they were almost entirely single-celled. Just coming from the East Coast to Madison, they seem to have quickly lost the ability. But every once in a while, I would see a multicellular one floating around, enough for me to realize that it still could do it, but for some reason wasn't in the lab.
And you were kind of interested in this question because this marks one of the major transitions into the sort of ancient transitions into the animal lineage, where you go from single cells like those ciliates that swim around in ponds to these multicellular guys that all the way out to complex mammals and so forth, right?
That's right. Yeah. When we want to think about the origin of animals, we really need to think about the origin of multicellularity. And this organism I could see, I had seen, was able to be multicellular. And here it is able to be single-celled too. So it seems that it can change from being single-celled to multicellular. And, one thing I should say is that the multicellular colony form, which we now call a rosette, it looks like an early animal embryo. It looks like, if you remember your embryology, it looks like the morulus stage. So that's the stage with about 16 cells. It's a ball of cells, no clear differentiation. It's just a sphere. That's what these rosettes look like. And so I thought, geez, if I could understand how these form, and if it proves to be related to what we see in animals, we could really learn something about how animal development first evolved.
Yeah, it's a cool idea. Yeah. So, is there, you kind of point this out in even in the, I think, abstract or the intro of this paper, that there's a difference between just cell aggregation versus multicellularity. What is that difference, or what distinguishes them?
Well, so I would say that the distinction is not, it's between development and aggregation, because, I think often people think a non-animal that's multicellular, they think about dictyostelium, in which you have a bunch of cells coming together, cells that can live on their own out in the world, and they come together and they aggregate into a multicellular form. But animals always go through a single-celled bottleneck. So all animals develop from a single cell, and that's the zygote. So the kind of development we see in choanos is that kind. It starts from a single cell, the cell divides, but now the daughter cells stay together.
Cool.
And those divide, and they keep staying together. So those are two different forms. There's the aggregative multicellularity that we see in dictyostelium, and then there's this clonal developmental process that we see in animals and choanoflagellates. And to be fair, plants also. So let's see, where was I?
Oh yeah, sorry. We keep interrupting.
No. No, that's all right.
So part of the backstory was you kind of already, before this paper, you were already onto some of the key. As you were saying, from the East Coast to Madison, you lost your multicellularity, but you sort of found the hidden accomplice, let's say, at some point.
So then at some point, this is after I started my lab at Berkeley, an undergraduate was treating S. rosetta cultures with different antibiotics to try just to clean the culture up so we could sequence its genome. And he had one cocktail of antibiotics that completely unexpectedly led to Rosetta becoming multicellular. And we had no idea what was going on, but eventually discovered that it was in fact the bacteria in the culture that were somehow triggering multicellular development. And we finally were able to track down, I say finally, this part actually went relatively quickly. We were able to figure out which species of bacteria, and it was the species Algorophagus, which had been co-isolated with the choano. So we know that it was actually out there in nature with the choano when it was first collected. So that's the backstory here, which is I had this organism, Rosetta, that we couldn't get to become multicellular, and then once we discovered that it required the presence of these bacteria, we were able to get it to happen whenever we wanted.
Yeah, that's interesting. And so the idea, I think, is that this, because it's a natural sort of co-occurring organism with the choanos, that there's something about its regular sort of lifestyle or replication phases that having these bacteria would trigger something like this, that would there be a fitness. Right. Yeah. And what is that?
Right. Fitness well, we don't know, frankly. Yeah. Uh-huh. We can, here's where we start hand waving. I think it's always, people want to have, stories about adaptation. But we don't really know what drove this interaction originally. So one idea would be that the bacteria are somehow, or let me back up and say that the rosette might be better at something, for instance, prey capture. So that would be a reasonable expectation. Maybe it's better at capturing bacteria. And so, if they could sense these particular bacteria that might somehow be preferable to be in a rosette. We've done some experiments. I'd say we have experiments that support both hypotheses. So depending on how we set it up, rosettes look like they're a little better at prey capture. Under other scenarios, they're no better than single-celled organisms or the single-celled S. rosetta. So we really don't know. And there are alternative hypotheses. So it could be that the bacteria are present at the same time as some predators of choanos. So there could be a co-occurrence, or it could be that they thrive in the presence of certain micronutrients that may, and we really don't know. So we're still trying to figure that out, but I try to move the conversation along when we get to that.
So fair enough. Remind me where S. Rosetta lives, and what fraction of the time there does it spend as single cells versus rosettes? Do we know that?
We don't know. And in fact, it's only been isolated once. And it was isolated from off the coast of Hog Island in Virginia, where there's a little marine station. We've gone back. We have the GPS coordinates, and we've gone back to try to re-isolate S. rosetta. And the exact place where it was isolated is now terrestrial. But students from my lab have gone to the closest water that they could find, and we've never been able to re-isolate it. So that's a little frustrating. But a former graduate student of mine is now working on the Terra Oceans project, which is a large project to identify the diversity and geographic spread of different microbial eukaryotes. And he sees signatures of S. rosetta actually in the South Pacific. So it could be that it was off the coast of Hog Island, but that's a rare event, and that it might be endemic to other regions that we haven't sampled.
So the ecology in the evolution sounds pretty complicated, but you have made a ton of great progress on more at the biochemical level, figuring out actually what's triggering some of this formation from the bacteria. So, and that was already known before this paper, correct?
Some of it was.
Some of it, yeah.
So, we collaborated with a really wonderful chemical ecologist, John Clardy at Harvard Medical School. And what we wanted to do was figure out what are the molecules produced by the bacteria that trigger rosette development. And this proved to be a problem that we couldn't solve on our own because it wasn't a protein. And once we get away from proteins, I'm completely out of my element. And so it became clear that the bacteria were producing something that they were releasing into the water, and that something was a lipid. And so the first stage of this process was to try to figure out what class of lipids could trigger this transition for S. rosetta, causing it to go from being single-celled to multicellular. And we nailed it down to a single lipid, a single class of lipids called sulfonolipids. And we named the first one of these Rf1 for rosette-inducing factor 1. So this molecule was sufficient to stimulate a small portion of the population to switch from being single-celled to multicellular. And if we didn't add this lipid, nothing happened. So the culture would be entirely single-celled. Mm-hmm. And so that was the backstory. Was that this very in retrospect it seems so small, but at the time to be able to go from discovering this phenomenon of bacterially induced multicellularity to actually finding one of the molecules, isolating it, and getting the structure was extremely exciting to us.
What's the time course? You add a compound to a single cell culture. How long does it take now for multicellular aggregates to form?
Yeah, so it's determined by the cell division rate. So it takes about 24 hours to get a 4-cell rosette, and that's the point at which we can tell that it's a rosette and not this other multicellular form that I haven't talked about called a chain. So once there are 4 cells, we can see that they're wrapping around on each other and they're forming a sphere. And so we add the molecule, and 24 hours later we can tell if it's a rosette or not.
Cool. So I guess that kind of sets the table for the paper today. So you found Rf1 a while ago, and after figuring out it wasn't a protein and isolating the lipid as this collaboration, but I guess that's not the whole story. It's sort of just the beginning of the story in some ways.
That's right. So we would of course love to find out if there's a receptor in the choanoflagellate. And so we were trying to work with Rf1 to see if we could find out what molecules it's touching in the choano, but Rf1 turned out to have all these problems. We would if we tried to tag it, it lost its activity. It has a very tight structure-function relationship, so if you change the stereochemistry at all, it doesn't work. But the biggest problem is that while it's efficient, it's not a very good inducer. And so, in fact, if we add live bacteria or conditioned media, we can get 80 or 90% of cells to form rosettes. But if we add Rf1, we're getting around 1 to 2% of cells in rosettes. So it's really a poor approximation of what the live bacteria are doing. Mm-hmm. So we initially thought, well, it's a lipid. The reason it's not fully active is that maybe it needs to be delivered in a membranous environment. Maybe it needs to be in outer membrane vesicles, or the choanos need to eat it. But a new graduate student, or at the time, new graduate student, Ariel Woznicka, really started testing, dissecting that hypothesis in a number of different ways, and ultimately was able to show that Rf1 its activity was not dependent on its delivery, and there really was nothing you could do to improve the activity of Rf1 beyond what we had from the isolated molecule. And so instead we realized, there have to be other molecules that the bacteria are producing that unleash the full induction of rosette development. And we weren't clear why we hadn't found them on the first round. But the answer, I think, clarifies that. So we went back. She developed a new bioassay. We again worked with the Clarity Lab, and now with this more sensitive assay, in fact, we did find one more inducing molecule, and its name is Rf2, and it looks like Rf1 in a lot of ways, but it's more potent. It induces a higher fraction of cells to form rosettes. And so that was exciting, but it also, like Rif1, did not recapitulate the full activity of the bacteria. So now we have these two lipids. They give us some rosette induction, but not a lot. And Ariel also noticed that the rosettes induced by these lipids were really small and kind of fragile compared to normal rosettes. So what she did was she said, okay, why can't we find all the activity? And she thought, what if the molecules have to act together? What if they're cofactors or synergists? So she went back and tested all of the lipid fractions in pairwise combinations at many, many different concentrations. So she titrated every single lipid fraction against itself. And when she did that, then she found the mystery activity.
So this is cool. Maybe I can just step back for a second. So you have this great assay for rosette formation. And then to get Rif2, and you had the first factor, but then to find the others, so you start fractionating out different lipids. Maybe tell us a little bit about how that process works, or what you're taking, fractionating, and then applying to pull out these other guys.
So okay, let me say that we know that live bacteria give us about 80 and 90%. And I think the fact that the bioassay is quantitative was very helpful. We knew that conditioned media would give us full activity as well. And so that meant that it was something that was getting released. And then the question was what class of molecule? And we knew that if we did a lipid extraction, that the lipids alone were, if you take total lipids, that also recapitulated the full activity of the live bacteria.
Gotcha.
So at that point, we could ignore proteins and other small molecules and just be entirely focused on the lipids. So then what we did, and by we, I mean the Clardy lab, is the royal we, was to take the total lipid fraction and now fractionate that. And it was fractionated based on charge, if I remember correctly. And actually on a variety of different parameters. So you take now your lipid fraction that's been separated in a variety of different ways into small subfractions, and now we can test each of those for its ability to induce rosettes. Okay, so there were two fractions that were eluded right next to each other that were active on their own, and those were the fractions that contained Rif1 and Rif2.
Gotcha.
And they were not very active, but they had some activity. We isolated the molecules, and it was Rif1 and Rif2, these sulfonolipids, which I should say are related to sphingolipids for the aficionados. And there was no other detectable activity in the lipids, in the rest of the lipid fractions. But what she did, and I can't remember the exact number of fractions, 14 or 15 of these, she then tested each of them in pairwise combinations with each other. And it turned out that there was a fraction that had no activity on its own. But when it was mixed with the Rif1 and Rif2 containing fractions, now elicited very, very high levels of rosette development. Okay, levels that rivaled that of the live bacteria. Okay, so that was really interesting. I should have the paper open in front of me so I can say which figure, but from memory, I don't know. But so there's a fraction, a little pool of lipids then that can't induce rosettes on its own, but when you mix it with the rosette-inducing factors, now you get a ton of development. And that fraction, that mystery fraction, contains what we are calling a synergist. It's a different class of lipids, and for some reason, when added in combination with Rif1 or Rif2, can elicit the full rosette development pathway. And that, I think, is really interesting because now we have, I
agree. So I think I have the paper open, and this might be figure 2C. So, and actually, I don't know if we've actually said, so the name of this paper is Bacterial Lipids Activate, Synergize, and Inhibit a Developmental Switch in Choanoflagellates. And so we're already at the activate and synergize phase here. And so it looks like figure 2C shows when you have the two fractions mixed, you start to see the percentage of cells in rosettes starts to kind of skyrocket. And then figure 4B, this now is again talking about this synergistic effect with the LPEs. So is this the LPEs? Right. What are those?
Yeah, so the LPEs, so once we had this little fraction that we knew could synergize with the RIFs, we continued subfractionating it until we had a pure molecule. Again, we being the Clardy lab. And actually, Alexandra Cantley really was doing the hard work here. So she was able to isolate two lipids. And it turned out that they were both lysophosphatidylethynolamines, and I had to practice that a lot to be able to say it. So, so we abbreviate that, as LPE.
Uh-huh.
And this is an interesting class of lipids. They are ubiquitous in eukaryotes. They seem to have some important signaling roles, and yet it's unclear how they actually work, what their receptors are, if there are receptors, what pathways they're stimulating. There's still a lot that we don't know about these lipids. And as far as I know, they've never been implicated in crosstalk between bacteria and eukaryotic hosts. And so it's really interesting that they seem to be involved in this case. And of course, once we see this, it raises the question of whether they might be mediating interactions in other host microbes systems that are harder to dissect at a biochemical level the way this one is.
Could I ask you, what was the starting point for, cause I guess it relates to the fact that these lipids are parts of membranes, so would they be expected to be soluble? Did you crack the bacteria open to get them? Are they secreted? Where'd they come from?
Right. So these bacteria produce what are called outer membrane vesicles, or OMVs. And so what happens is the bacteria produce excess membrane that gets blebbed off. And there's a lot of interest in whether that blebbing process is unregulated or whether the OMVs or the outer membrane vesicles are actually delivering signals between bacteria or even between bacteria and eukaryotes. So we don't know why they make OMVs, but we do know that the rosette-inducing molecules are contained in OMVs. And so that's when I say soluble, it's miscible. They're
right.
Yep.
They're still in these these little tiny membranes. Yeah. So that's the synergizing part, and it turns out that not only do the LPEs unleash higher levels of rosette development, but they also seem to drive a maturation step in rosette development that we didn't previously realize existed. And so if we're going to refer to specific figures, this is figure 5. And what my graduate student Ariel realized was that when she treated the choanos with the LPEs plus the rosette-inducing factors, now they were full-sized, they were robust to shear forces. They just looked exactly like the rosettes that are formed in response to live bacteria. And so we hadn't appreciated that there might be at least 2 stages at which bacterial cues are necessary in order for fully formed rosettes to form.
That's cool. So, and we're looking in that figure, there are these great images of the rosettes themselves, I guess, just light microscopy images. And there's also this, in one of the rows, so you're showing tubulin staining to get a kind of an outline of the cells. But then there's also this channel rosetless. What's that?
Right. So it's very exciting.
Yeah.
So Rosettas is a protein. It's a C-type lectin protein, and it has proven to be. Here we're using it in a relatively boring way, which is as a marker for rosette development.
Mm-hmm.
If we only see this protein in rosettes, but our ability to use it really is a result of some incredible work by another former graduate student, Tara Levin, who from scratch developed a way to do forward genetics in S. rosetta. And so she first of all, she discovered the sexual cycle in choanos, which had never been observed before. She then developed a way to do mutagenesis and to screen for phenotypes. She screened for mutants that were defective in rosette development, and she mapped one of those mutants to this gene, Rosettas. It's very exciting. So this is the first gene that has been implicated in rosette development, and it's very exciting. And here, its only show in this paper is as a marker. But the reason we're showing it is just to demonstrate that these groups of cells, for instance, in panel D on Figure 5. You might say, well, maybe that's a rosette, maybe it isn't. But the fact that it has rosetless in the middle says the cell is at least, or the cells are at least trying to form a rosette in that case.
So you do not see rosetteless if, for example, you add Rf1, 2, and Lpe, and just before rosettes form, you don't see it being produced on the surface, right?
It's at extremely low. So in cells that have not been induced, it's there at very, very low levels, and it is not secreted.
But if you induce them, you don't see it before rosettes are formed, right?
You see it, like, I would say actually within short order, you would actually see it upregulated and secreted, even before the first division.
Okay. I should say that this paper is an open access, so if anyone wants to follow along, you can do it. You don't have to have any fancy subscription.
Yeah, good news.
That's right.
And actually, just as another side note, so Nicole's former student, Tara Levin, you can now find her up in Seattle as a postdoc in Harmeet Mullick's lab.
Wow.
The same lab
That's
where
right.
I spent a few years.
Small world.
That's right.
Yeah.
Yeah.
Okay, so we have all of this activation and even synergy, which then, as you mentioned, sort of revealed a new maturation step that you hadn't even realized existed. But then it seems like the story takes another twist, which is that you actually found an inhibitor of this process. So how did that come to?
Right. We were not looking for an inhibitor, but as Ariel started mixing all these lipid fractions together, she realized that there was another fraction that had no obvious activity on its own, but when mixed with the rosette-inducing factors, blocked rosette development. And this was quite unexpected. And when our collaborators in the Clarity Lab isolated this molecule, it turned out that it actually looks like it might be acting as a competitive inhibitor with the rosette-inducing factors. And that's not only because of this biochemical property if you mix them and it doesn't work, but if you look at the structure, and this is in Figure 3C, you can see that it looks like a hydrolyzed form of one of the rosette-inducing factors. The structural similarities are striking. So now we get back into the hand-waving evolutionary realm, which is what's going on here.
That threw me for a loop, because if I was just sort of thinking, you brought it up earlier, this idea of sort of predator-prey relationships, right? But then if that were true, the enhancers and synergizers might make sense, but then the inhibitor is sort of a curveball.
Right. Well, so one might be inspired to say, well, maybe the bacteria are making the inhibitor in order to block rosette induction. I just don't know how to think about all of these things. I think these lipids are all being produced as part of the normal lipid metabolism of the bacteria. Yeah. And the choanos seem to have evolved some means of eavesdropping on these molecules that are being released. You then argue, then the bacteria must evolve some way of getting around that. And what's unclear in terms of when we think about microbial ecology is how strong the selection is. Because I don't know how often they're encountering, how often are the choanos eating Algoriphagus, for instance?
Yeah, right. Yep.
They're not selective. So I just don't know what's going on. I don't know.
Yeah.
But you don't think they have to eat bacteria to acquire these stimulators and inhibitors, right?
No. So we can isolate the outer membrane vesicles from the bacteria, and they're sufficient.
Yeah.
And if you look at the levels of lipid that are required to induce, they're quite low. So we don't think they have to eat the bacteria.
But the idea, you really favor the idea that there's a receptor for these lipids and that the inhibitors are somehow competitively binding and inhibiting in that way, right? So I guess that's something you'd like to go after, correct?
Right.
Yeah.
And that's where the genetics comes in. Ideally, we'd like to take a biochemical approach. If we could tag the lipids and then use tagged lipids to find the receptor, that would be fantastic. But so far, that hasn't been straightforward. We lose activity. I think this inhibitory molecule has some potential, but that hasn't worked out.
You know what you could do? You could take one of these lipids and conjugate it to something that's toxic, and then select for mutants that are not killed by it.
I like
cool
that.
idea. Yep.
That's a great idea. Yeah.
With the genetic
we'll do that.
Well,
that's a great idea.
Don't...
I guess one challenge. But they're all. I think, regardless of what their structure is, they're all going to get integrated into the choano membrane. So if there's some way to make it be toxic only if it binds to its receptor. Yeah.
One of the things this got me thinking about with some of these really cool open questions. I mean, so you draw some analogies to, in I think the discussion, a bit about how, we know of other systems where bacterial cues are actually really important to eukaryotes. So some of the symbiotic organs or vesicles that form in some eukaryotes, like the bobtail squid,
right?
But also the immune systems and how sort of they train on cues from the microbes that are associated with the hosts. And so, I mean, one system that we sort of tinker with here is this idea of the cyclic nucleotides that a lot of bacteria build and use as secondary messengers to communicate. And this is thought to, be involved in whatever biofilm formation or other bacteria to bacteria sort of communication systems. But what's happened is our immune system, in a sense, has listened in, and we now have examples of enzymes that build very similar cyclic nucleotides or modified nucleotides that then act as second messengers or triggers to signal our own immune system. And so I'm wondering, is this possibly happening in choanos as well? Are there any staphanolipids or other similar lipids made by the choanos?
So they do make sphingolipids, but they don't make any sulfonolipids as far as we can tell. And we've never seen them form rosettes in the absence of bacterial cues. So you would expect that they should have some way of stimulating their own rosette development, but we have not found that.
Yeah. Right. I'm wondering if there are properties of the sphingolipids from the choanos that might be mediating other processes, and this is sort of almost like a echo or crosstalk of, some ancient signals or cues.
Yeah. It's possible. We haven't seen it though, not yet.
So I guess there are many questions, but one I suppose you think about it a lot is does this actually happen in nature?
Right.
And given that you can't find these, that's a tough one to answer, right?
Yeah. I mean, all I know, I suspect it does because we isolate it from nature as a multicellular form. So we knew it, at least at the moment of isolation, it was multicellular. And we know that the Algoriphagus bacteria were there at that moment. Right. And so at least once it was the case. We, whether it's one possibility, in fact, let me back up and say we've been testing lots of other bacteria for their ability to elicit rosette development.
Right.
And it turns out that, in fact, there are a lot of other bacteria that can do this. So it's not a generic property of bacteria. Most bacteria can't, but there are a lot that can. I think we have at least 20 or 25 other species. And they are not all Bacteroidetes. Some of them are gram-positives. Some of them are members of the verruco microbiota, which is a new, relatively poorly understood group of bacteria. And some of these other species don't produce sulfonolipids. So the choanos are, we assume, responding to other classes of molecular cues in those cases.
Yeah, interesting.
So I suspect that maybe the specific cues from Agoraphagus are not, that's just one way in which rosette development can be induced. And it seems that there are a lot of cues that can get this rolling. And we're just now beginning to figure out what some of those other cues are.
I guess that makes sense that it wouldn't depend on just one bacterium, right?
Right. I don't know.
Although there's certainly, the bobtail is one bacteria that are recruited, right?
Right.
And there are other examples of, I think there's a caterpillar that Joe Handelsman works on. They have its microbiome consists of 2 different species, so.
Yeah.
Right.
Well, there are certainly many examples, I think, of, endosymbionts where that sort of evolutionary relationship has been built up over a really long, and the physical association is so close that those do become pretty specialized. Yeah. But this is interesting because you have almost this more long-range communication or signaling going on. And so the fact there are so many inputs and possible outputs seems really cool.
Yeah. Well, I suppose if you go far enough in your study of this system and, you look at the genetics and provide lots of compelling evidence for a receptor and so forth, that it's likely that it happens out there. And it's one of those things you may never be able to prove.
Right. Well, this discovery that they seem to be living in the South Pacific too, I think I can somehow motivate myself to go look for them there.
That's not a bad place to go.
Good idea.
Hopefully we can find some in nature and get a better idea of what's going on out there.
Yeah, that'd be cool.
So, and maybe we can return to this, one of the questions that we sort of started with, or one of the ideas we started with, which is using this as a model for potentially some of the key evolutionary transitions from single cells to multicellularity. So how do you think that connects to perhaps our direct ancestors that then led to our lineage? Do you think this has some important clues to how that might have happened?
I can tell you how it's changed the way I think about it. I would not argue that these specific classes of molecules stimulated the transition to multicellularity. And I wouldn't even go so far as to say that bacteria did. But what it has forced me to think about is the fact that the first animals did evolve in oceans that were already dominated by bacteria. And I think it's a reasonable inference that the first bacteria made a living by eating bacteria. The first animals made a living by eating bacteria.
Yeah.
So all of this has made me, I think, forced me to try to take the bacteria into consideration more when thinking about animal origins. It's certainly, I think we have to think about the possibility that bacterial cues did contribute to the regulation of biological processes. But I think it's really premature to think about what particular processes they might have been influencing and how they might have done that. And, until we can find some evidence for conservation between choanos and animals, I think it's a black box. And given how quickly host microbe interactions evolve within animals, I think the likelihood is that there's really not going to be a strong signature that's going to convince us of conservation.
Yep.
So
that's really
time will tell.
Really fascinating. Lots of cool adventures ahead for these projects.
Yeah, it's really fun. And I should say that there's more to come. So I'll do a little teaser that we've discovered another really exciting process that's regulated by bacteria in choanos. It's sex. They can induce mating with a different class of bacteria. And we've isolated a new choanoflagellate species from Mono Lake, and it seems to be farming bacteria inside of its colonies.
Oh, wow.
Yeah. So I think it's really early days, but I think that choanos are living intimately with bacteria. They eat them, they're surrounded by them, and they seem to be particularly attuned to cues that they're producing, perhaps as some way of interpreting the environment in which they find themselves. And so, I think maybe the first animals also might have used those types of approaches, but we really don't know until we start looking more at sponges and Cnidaria and ctenophores.
It sounds like what we do. We're eating bacteria. We're surrounded by them. They make us behave in certain ways.
That's right. Mm-hmm. That's right.
How many labs in the world work on choanoflagellates?
So there's my lab, and then there are a few labs of people who came from my lab. Mm-hmm. So Rosie Allegado, who's at the University of Hawaii. Pavel Burkhardt, who's now at the Marine Biological Association in the UK. And Scott Nichols is at Denver University, and he's studying sponges and choanos. And then there are a number of labs that they're not solely focused on choanos. They study choanos as part of a larger research program. Primarily, they're involved in microbial ecology and thinking about, they are looking at microbes in the environment, and are largely less focused on molecular and cell biology. So we have if anyone's interested, we have a choanoflagellate workshop every 2 years. And we typically get around 30 to 40 people at these meetings. So it's a small but hopefully growing community.
Is there a website for that workshop that I could direct people to?
Yeah, is it on your? How about tomorrow? Well, we're just setting the date. It's actually going to be the week of May 31st, 2017. And I have tweeted about it. And we don't have a website yet, but we'll get one up within the next few days.
Okay. Maybe you could tell us the roles of the people on the paper that we just talked about.
Sure. Yeah. So let me, okay. So Ariel Woznicka is from my lab, and she she studies the choanoflagellate side of this interaction, and really did a lot in terms of thinking about how would you go about designing a bioassay that could help you find the missing activity. Alexandra Cantley is in the Clardy lab, and she figured out how to isolate these molecules. And Tina Biemelmans helped with structural analysis and was involved in helping to characterize Rf1. And Elisaveta Frankman helped with some of the structural analysis. And then John Clardy and I are the senior, what are we called? The grizzly on gorillas. But he and I have just had a wonderful collaboration. There's been a lot of, thinking about how to do this and how to figure this out. And he's been a really great sounding board for thinking about how to interpret the ecological significance of this interaction. It's been wonderful. Yeah.
That's great. And I guess the first author, it's co-first authors, I guess, reflecting kind of, on both the Clarity Lab and the King Lab sides, the contributions.
Yeah.
Yeah.
Well, that's a great story.
Yeah,
really interesting. We really appreciate your joining us today. Nicole King is from the University of California at Berkeley. Thank you so much.
Thank you. This was fun.
All right. Bye-bye.
Yeah, thanks, Nicole.
Okay, take care. Bye.
Yep. Bye.
I'd like to tell you about one of the sponsors of this episode of TWiEVO, CuriosityStream, the world's first ad-free nonfiction streaming service. They have over 1,500 titles, 600 hours of content, so you could binge watch if you'd like. How many days? That's a long time. 600 hours. You'd be watching for a long time.
Yeah, I'll tell you what, my curiosity got the better of myself, and so I went to CuriosityStream finally, and my goodness, it's just it's chocked full, as you're saying, hundreds of hours of high quality, really good stuff.
Yeah.
David Attenborough's all over the place. It's just It's like a treasure chest.
Did you find Neil Shubin? I bet he's on there, right?
He must be somewhere in there, yeah.
It was founded by John Hendricks of Discovery Channel, and that's why you got real science shows, not reality TV shows that you find everywhere else. So for the listeners of TWiEVO and all the Microbe TV podcasts, this is probably something you'd like. Mm-hmm. And you have other things besides science as well, but you can watch it on the web, on your appliances like Roku, Android, iOS, Chromecast, Amazon Fire, and Kindle, Apple TV, 196 countries. They have a lot of science and technology. They also have nature, history, and a lot more. But the common theme is it's all nonfiction. There's nothing fake about it. Library includes Stephen Hawking's Universe, where he traces the history of astronomical theories and technology. Deep Time History, a 3-part series that tells the story of the history of the universe. It's 14 billion year history.
Yeah.
Some of these are single pieces. Some of them are multiple piece sets. They have Underwater Wonders of the National Parks. This is the 100th year anniversary of the National Park Service, and this 7-part series takes you underneath the water within national parks of America. Cool stuff.
Very cool. Yeah, I agree.
They have one of the largest nonfiction 4K libraries on the internet. They have over 50 hours of 4K content, which of course is ultra high definition. They have monthly and annual plans available starting at $2.99 a month, less than a cup of coffee, or a single title on some of the competing platforms. So check out curiositystream.com/microbe. Use the promo code microbe during sign-up, and you'll get unlimited access to the world's top documentaries and nonfiction series, completely free for the first 60 days. It's 2 months free of one of the largest nonfiction 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign up. We thank CuriosityStream for their support of TWiEVO. TWiEVO in Vivo. It's coming up, huh?
Yeah, next month I'll be in your neighborhood, and so we will be back in Vivo for our second installment.
Nice.
Yeah, I'm looking forward to that. Yep.
Let's do some email. You know, we've accumulated some emails since we last spoke. The first one is from Anthony. There was a mention of living fossils in the recent TWiEVO. Attached PDF is a paper about ants in amber found in central New Jersey. Your photo starting at page 10. The fossils are 90 million years old. Something very similar might still be seen today in any backyard. The Hadrosaurus, followed very much later by mammoths, were definitely more impressive New Jersey residents. Both are very long gone. What might it be about the ants that allows for their continued success, whether dinosaurs, giant mammals, or trucks dominate the landscape? The seemingly changeless nature of ants perhaps is what prompted Dali to feature them frozen in amber in persistence of memory.
I love it. There's also one of my favorite Dali images is this photograph of him walking a anteater, an aardvark, outside of the Paris subway.
Cool.
And makes a statement about maybe picking a high maintenance pet and sort of the artist's interpretation. It's actually kind of an interesting idea. Yeah.
So this paper is in it's published by the American Museum of Natural History. It's called American Museum Novitates. And that's the museum here in New York City. It's called New and Rediscovered Primitive Ants in Cretaceous Amber from New Jersey.
Yeah, I took a peek at that. There's some interesting stuff here. I would make maybe a couple of quick comments. So first, this idea of living fossils, I think I always try to be a little bit careful with that phrase. We've probably been throwing it around a little bit loosely. But certainly these are, like all fossils, dead ones, preserved in amber. And it is true, these guys look like, to some degree, I think Anthony's point, they look like ants of today, at least to our eyes. But what I'd be curious, actually, I was thinking about this, is whether if an ant looked at one of these 90 million old ants, would it look quite different to it? Or would it smell different? Or would it have different behaviors? I mean, from an ant's eye view, there might be quite a few changes. Yeah.
I'm sure this is going on, but aren't people pulling out DNA from these and sequencing them?
I don't know if you can go back that far to 90 million years. There are much newer amber samples where that is happening.
So these are too old to have DNA, is what you're saying?
As far as I know, 90 million years is a long...
It's interesting that they have a nicely preserved ant structure, though, right?
Oh, yeah, the ants themselves look beautiful. Yep. Yep. And then in terms of, I guess, why are the ants the ones that are still with us, but not the mammoths and the Hadrosaurus? I think the insects have proven to be a pretty resilient and, they've survived a lot of insults where some of these bigger organisms, like the mammoths, for example, might be more vulnerable to environmental changes. Yeah.
Well, you need a pretty big piece of amber to preserve a hadrosaur.
Point taken.
Anthony continues, your speculation of conquest of the Neanderthal along the lines of the fall of Troy certainly is as good as any other. I guess that was something we talked about when we did the Neanderthal paper, right?
Correct. Yeah, and we were getting into a little speculation about how they might have been wiped out by our own species. Definitely speculating.
Yep, for sure. On TWiEVO, Professor Despommier has posited that Neanderthals were exterminated by our diseases. I'd wonder about the climate change of the time. This definitely enabled the spread of Homo sapiens. Why not other organisms from Africa? These may have been vectors of diseases that our species grew up with. The Ice Age specialized Neanderthal would have had no resistance. Yeah, it's possible, right?
Possible. So maybe in this analogy, we're more like the ants than the mammoths.
Yep. Want to take the next one, Nels?
I would. So Justin writes, Dear Nels and Vincent, I'm loving this new podcast. I'm a postdoc at Texas A&M University. I just finished my PhD work in Utah, and listening to Nels talk about Salt Lake makes me feel somewhat nostalgic and homesick. Now we're talking. The University of Utah really is a fun place. I'll second that, Justin. I have a question about TWiEVO episode number 8. I may have missed something, but early on in the episode, there was some conjecture about the interspecies interactions that resulted in Neanderthal genes making it into the human genome. Here we are again, echoing off of Anthony's points. Some of these conjectures assumed that rape, kidnapping, and the like may have been the primary drivers of this interspecies mixing. Is there any way to know, either by looking at sex chromosomes or even mitochondrial DNA, if the Neanderthals that contributed to our genome were predominantly male or female? If we knew the answer to this question, it would give us some very good hints about how this all actually happened. I also have a listener pick. Should we save that for the listener picks, or
sure.
Might as well? Yep.
Yeah.
Okay, we'll save the we'll save the end of Justin's note for a minute here. Yeah, boy, I think we're again on this sort of somewhat speculative ground. I think it would be it's an interesting idea. I think just looking by sort of sequence gazing at either sex chromosomes or mitochondrial DNA, it might be really hard to see sort of evidence of some of this speculation, just because there's been so many generations and turnover of mitochondrial genomes as they've gone through. The potential clue there is that goes exclusively through the female lineage, since the mitochondria is contributed through the eggs and not the sperm.
Right.
But to kind of be this far along and knowing how actually little of the Neanderthal genome is still sort of proposed to be introgressed into our own, those traces like that might not be so visible, right, at least from my impression. Yep. Good idea, though.
Yep. Hey, they train them well in Utah,
There you go.
We're back. Another one from Anthony. He sends a link to an article. There's a mural of Darwin on the outside back wall of Avondale Wine and Cheese in Alicia Alley, which is somewhere in the UK, I presume. Mm-hmm. And it's been restored because over the years it was trashed. Mm-hmm. On Friday night, someone vandalized one of West Ashley's most beloved works of art, a Darwin's Secret. It's a picture of Darwin with his finger, saying shh.
Mm-hmm. Yep, famous image.
Created 4 years ago by local visual artist Sean Williams, and it was mutilated, so then they restored it. So it looks very nice now.
Yeah, looking at it. I wonder if Darwin, how he would feel about that. Maybe the, vandalism, is that just sort of the next artistic selection of the, sort of next artistic generation? I don't know. I think I'm probably overthinking it.
Yes, we tend to do that, us scientists. So he said this is another image brought to Facebook by Kim D'Alessandro. Kim D'Alessandro was the wife of Joe D'Alessandro, who was one of Andy Warhol's, I don't know what you would call him, right?
Yeah.
He hung around with him. But she had put up a picture on Facebook a while ago of embroidered Petri dishes, which totally went viral. They're just beautiful. They're made of embroidery, but they look really beautiful and they're fuzzy.
Yeah, that's cool.
And now this is another cool thing. Okay. And we have one here from Trudy. Dear Vincent and Nels, during my daily perusal of the Internet for Cool Science, I came across an editorial in Nature which talks about modern gene editing tools and how they're used to understand the mechanisms of evolution. Of particular interest to me was the attached paper by Nakamura et al. Apparently these guys and gals used CRISPR-Cas9 to inactivate the genes involved in zebrafish development, producing animals having fin tips more like the feet and digits of land vertebrates. I can already hear the anti-GMO crowd and creationists chomping at the bit. Might make for an interesting snippet for a future episode. Thanks for all you do. Yeah, this is a paper by Neil Shubin.
Yeah, I know the paper. It made a big splash maybe in the last few weeks even. I think it was the cover A1 article or page A1 on the New York Times for in the print edition. So it was a good sort of victory for science getting out there. I know the story a little bit, so I've seen Neil Shubin in person speak about it a couple times now. And it's really, I think it's inspirational on a couple levels. One is, as Trudy points out, using some of the newer technologies for gene editing in order to actually do these sort of functional tests, in this case of development. In this case, what I think is really inspiring is how Neil Shubin has gone all the way from the paleontology to the molecular biology. And actually we had echoes of that with Nicole today. She, as she mentioned, she trained as a paleontologist for a while at Harvard. We also have an echo of that from a previous guest, Mike Shapiro, who's now doing the pigeon genetics and genomics here at Utah. And so this is, again, I think it's a really fun sort of example of how people are really taking these broad swaths of science all the way from the really ancient literal fossil records, the dead fossils, to experiments to try to understand that evolutionary process. Neil Shubin and his work is a great example. Nicole King, I think that we saw today, another really good example, as well as Mike and many others. And so another exciting example of this functional synthesis of evolutionary thinking all the way through molecular biology. And I'm sure we've highlighted some of Neil's Shubin's stuff as I think some of our picks, and I'm sure we'll be hearing from him again, and hopefully as a guest at some point as well.
I think we'd get him on, and we'll certainly talk about this for sure, right?
Yeah, absolutely. That would be really fun. Yeah.
All right, last one's yours.
Okay, Jeffrey writes, Doctors Elde and Racaniello. This episode inspired several questions. I think he's referring to our recombination is for the birds, the PRDM9, in the story from Molly Przworski's lab at Columbia. Unfortunately, this seems to have been a case where knowing how to ask a question is important. I was only able to find partial answers to my questions and was hoping that you gentlemen might find them worth asking as well. These questions are kind of long. If you do use my questions on an episode, then feel free to use only the ones you have time to use. I think Jeffrey will just go through them and see what we can do. So number one, you mentioned that PRDM9 is known to be responsible for some forms of mammalian sterility. Might it also be responsible for lowered fertility? And so just as a little bit of background, so PRDM9 is this gene that's involved in meiosis and sort of marking or interacting with what we've discussed as recombination hotspots. So areas of the genome where through meiosis recombination is more likely to occur. And then some of the interesting work that we covered was this idea that maybe some organisms like birds may or may not have these recombination hotspots. And then when it comes to PRDM9, this idea that the organisms that do have this gene, that it's been known, it was first discovered as a so-called speciation gene, something we've talked about on some previous episodes as well, that as the gene code changes, it can do so in ways that if you hybridize or make hybrids of two individuals that have different variants of a gene, that they no longer are fertile. And PRDM9 was implicated as one example in mouse in particular. It's actually, I think it's still the first and maybe only mammalian speciation gene. A lot of the others that are known have been studied in Drosophila and a few other organisms. So Jeffrey is talking about PRDM9 and this idea of whether it's not only potentially responsible for sterility, but also lowered fertility. So, and then he says, this is the question that I was able to answer because I figured out that I was asking the question at the wrong point in the fertility question. I was wondering if recombination mismatching might be causing reduced successful fertilizations in some couples that wouldn't have occurred if they were coupled with different partners. However, I realized that the answer that you gave on the show was pretty much the answer as far as PRDM9 goes. PRDM9 operates at the meiosis stage. Thus, any PRDM9 effect should result in a defective haploid, especially sperm, and lowered fertility to sterility with all partners rather than partner-specific reduced fertility. So yeah, I think that's about right. It's from my reading of this as well. So if you remember, so the idea here is that kind of the stage at which this reduction in fertility or in sterility sort of matters for thinking about speciation. So in this case, you have two parents, the mother and father, each contributing a variant of PRDM9. And the idea is if that variant is different enough, that then the offspring of that mating, that's the one that's sterile. And so somehow this mismatch of PRDM9 alleles, this quote-unquote speciation gene, that's sort of the source. And so then if the offspring is already infertile, then that sort of answers the question about, Okay, that's how many do we have here? Oh, there's 3 more. So the other ones are a bit shorter. Number 2, have there been studies on the genetic phylogeny of the origins of PRDM9? I found a partial answer for that. This is Jeffrey going on. I didn't find any information on PRDM9 prior to mammalians, but I did find that it occurs in Mesotherian and non-placental mammals, specifically platypuses. Perhaps, as one might expect, platypus PRDM9 is significantly different from any other extant eutherian PRDM9. However, I was still not able to find out theories or evidence on the origin of PRDM9. Do you know what the leading theories are? Was it a radical repurposing of pre-existing zinc finger system? Was it a gift from a retrovirus? So just to maybe readdress the first part. So that is true that if you look in many lineages where PRDM9 can be found, that there's also rapid evolution, not only in the rodents where it was first kind of discovered, but also in primates, actually. So Harmeet Malik's lab, in collaboration with Chris Ponting's lab, had a paper maybe about 6 or 7 years ago. Actually, our guest Nitin Fudness was an author on this, where they looked at evolution of PRDM9 in primates, even in humans. And there are signs of rapid evolution there, just as there is in rodents and probably in other lineages as well. I think the origins question is a bit harder, and I'm not aware of people who have really kind of fleshed this out. So what is clear, I had to do a little bit of, I'm just pulling up a website here to try to get a little bit of background or just to refresh my memory. So PRDM9, this is, again, that's, the shortened acronym for a gene. It's PR domain zinc finger protein 9. And so what it looks like is sort of a fusion of a few different functional domains. So one is the protein has a histone trimethyltransferase activity domain. It also has a CRAB domain. This is another, if I'm remembering, a DNA binding activity, a Kruppel associated box. These are known for repressing transcription, so binding to DNA. And then the C2H2 zinc finger domains as well, which are also DNA binding. So I'm just taking a guess, but so you could imagine that just as sort of genomes are constantly or genes are sort of moving or breathing, that they could have come together in sort of a new functional way from a more ancient zinc finger. It's certainly known that some of these zinc finger domains are perhaps involved in curbing the activity of retroviral elements, either endogenous elements or selfish genetic elements, so things like lines and signs. And so perhaps there is some echo of a gift from a retrovirus, at least indirectly in that sense. But it's an interesting question, and I don't know how much progress has been made on that. 2 more to go. So number 3, question 3, you mentioned that meiotic recombination mutation rates are lower than the theoretical limits, suggesting a control mechanism about which we know little or nothing yet. Is there any evidence that this mystery control mechanism could become deregulated under certain types of stress on the organism. If this happened, then it could allow for higher mutation rates, a form of hypermutation, and thus potentially faster genetic adaptation in times of environmental stress. So this is an interesting question, Jeffrey, and it's a bit controversial, this idea of kind of adaptable mutation rates. There is some evidence for this. I think the most recent paper I can remember was work from Nadia Singh's lab. She's at NC State, although I think I heard her lab might be moving to the University of Oregon pretty soon in the next year or so. But anyway, her lab, she has a paper looking at Drosophila. And if I'm remembering correctly, they used thermal stress, turn up the temperature on the flies, and actually measure higher rates of recombination. And so this provides some empirical evidence for that idea. It's still a bit controversial. And so I would actually, if you're interested, I think it's an open access paper. And that might be worth digging in on Nadia Singh's work in that department. She also sets up sort of, I think, the controversy in terms of what was previously suggested, and then they weigh in on it with their study. Okay, and now the last one, number 4. In a more general question about sex and meiotic recombination, is there a single main control system for this process across kingdoms? Have different mechanisms for meiotic recombination arisen independently? Have we been able to develop a genetic phylogeny for meiotic recombination systems, and do we know how they arose? So I would say I'm doing a little bit of speculation here, but if my memory serves correctly, that meiosis, this sort of fundamental process in a lot of eukaryotic reproduction, possibly arose once. It's certainly very highly conserved. A lot of the proteins that you see, for example, in metazoans and in our species, all the way out to, for example, yeast, where meiosis has been well studied by a number of labs over the last 30, 40 years. And so I think this is a really, and in that sense, a highly conserved process. What's interesting is there have been all of these kind of interesting cheating events that have potentially arisen to try to kind of almost game meiosis to the advantage of one chromosome over another. So if you remember, in sort of the reductive meiosis process in females, in mammals in particular, you have you start with 4 chromosomes and you end up with 1 in the oocyte, or effectively 2, given that it's half, given that it's a reductive process, 2-step process. And so just from a selfish gene perspective, there's a massive evolutionary incentive to basically be the chromosome that makes it through the sort of chromosomal gymnastics of meiosis. And so there are a number of systems where this has been studied. I'm thinking of monkey flowers, Drosophila, other species. This is a topic, meiotic drive, that's studied up in Harmeet Malick's lab as well, as a genetic conflict where you get these sort of rapidly evolving systems on top of a fundamental genetic process. And so that might get at some of the question. I'm not going all the way back to how meiosis arose. I think that's another interesting question. I don't know much about that, but yeah. So then Jeffrey closes with thanks for the thought-provoking show. And I would just say thank you, Jeffrey, for the
great question.
Thought-provoking questions. Yeah, really good. Yep.
I want to tell you about the other sponsor of this episode, Drobo. I think this is the first Drobo spot we're doing here on TWiEVO, right?
Yeah, that sounds right. Yeah.
Drobo is a maker of expandable yet simple to use storage arrays for your computer. It was designed to solve 2 problems that affect hard disk drive. First, hard drives get full, and second, hard drives fail. And Drobo solves these problems. Both of these problems, because it's expandable, they consist of a small box with slots in which you slide hard disks. You can have from 3 to 12 bay Drobos, whatever you want to buy. When you put a couple of drives in, they're recognized and pushed together as a single drive, and it shows up on your computer as one drive. It's sort of like RAID, but it's the Drobo's own award-winning beyond RAID technology. You can put different size drives in. Okay, so it's expandable. You can keep adding drives. It's data aware. It knows how much space is available on your drives. It has green lights on the front. When they turn orange, your array is full or getting full, you put another drive in, and it's immediately incorporated into the array. Of course, when it's red, you're full and you have to do it immediately. So it knows how much space you have. Most important thing is probably redundancy, right? If you put 3 hard drives in and you copy data to it, it's actually duplicated across all 3 drives. So if one of them fails, you can just take it out and put a new one in. You don't lose any data. It's redundant. It's very cool. Go ahead.
I was just going to say, I can speak with some experience using the Drobo. So you were nice enough to forward along a Drobo that I think was supplied by that crew.
That's right.
So, and we were running into the first problem of the 2 that you mentioned, which was hard drives getting full. And so we've been doing this new, relatively new form of DNA genome sequencing, these Oxford nanopore MinION systems. And so basically the way this works is you hook a little device about the size of a harmonica through a USB drive into a laptop. And then you squirt a little DNA into the device, and it starts generating massive amounts of sequence data that takes up space on a hard drive.
That little harmonica thing is actually doing the sequencing?
Correct. Oh yeah, I have a photo. A photo here. Yeah. We'll put this up on the website. Wow. So we have you'll see we've got little stickers on the harmonicas, the Oxford nanopore MinION. So we've named one of them LeBrian James, and the other I think is called Ion Maiden. Those were our 2 sort of 2 device names. And so yeah, that's a DNA sequencer, genome sequencer. And in the background of this photo, you can see we're using it to sequence vaccinia virus genomes, and actually it's working beautifully. We'll hope maybe at some point we'll be able to talk about some of this data. But what you can see in this image that we'll put up is a Drobo in action. You can see those green lights of the 3 hard drives, which are absolutely starting to get filled up with sequencing data. And this helped us out of a jam. We ran one of these one night, came back, and the hard drive was full, and so we hooked up the Drobo. And we haven't run out of space since.
So this little MinION is attached right to the Drobo, is that correct?
It's actually not. So the MinION is attached to a laptop through the USB, and that's sort of off of the picture. And then you can either do a direct kind of Ethernet style link to the Drobo or Wi-Fi, which is what I think we were doing in this case. And so the way it works is even more slightly more complicated, which is that the all of the data that comes in off of the sequencer then gets sent up to the cloud, where the Oxford nanopore computers in the cloud sort of decode what the electric signals are in terms of what it's called for the base calling, and then that gets sent back down. And so there are 2 stages where you can start to build up locally the data onto your hard drive, and so that's what we were able to get around with the help of the Drobo.
You have that plugged into a little switch there, I think. Is that right?
Yeah, exactly. Where we have the Ethernet.
And so it's network accessible, right?
Exactly. Yep.
So
and then you could also use it to back up the second problem for keeping your data either redundant or backed up. We actually have plenty of space. We could do that as well.
So next time we'll tell you about how Drobo is having apps written by third parties so that you could put your Drobo on the world's network. So you could access it from anywhere, not just from within your lab or within your home. But if you go to a meeting in Naples, you could access your Drobo. You can make your own Dropbox, basically. But it would be a huge Dropbox because you could put lots of drives in there. But the key point here is that it's simple. If you can read a traffic light, you can use a Drobo, green, orange, and red. They make a number of products, 8 different products. Which one you want to get depends on what you want to do. If you're a photographer, you want a 5D or 5DT. These are 5-bay Drobos with high-speed connections. They have portable models that are about 2 pounds that you can hook up in the field. If you need a huge amount of storage, they have 3, 5, 8, or 12-bay models you can use for backups and so forth. And they have this network-attached model, the Drobo 5N, which Nels got, which I also have here, and I want to use in my lab to share data. And as I said, next time we'll tell you about how they're writing apps to share it even broader than your local laboratory.
Yeah, sounds good.
TWiEVO listeners can save $100 off on their purchase of a Drobo Mini Drobo 5D, Drobo 5N, or any 8 or 12 drive system. You just go to drobostore.com and use the discount code Microbe100. I think for labs, this is a great solution, especially with all this sequencing data and other data as well. So check it out. We thank Drobo for their support of TWiEVO.
Yeah, thank you. Thank you for helping us out with our data issue here.
Yeah, I figured you want to use it. You know, the guy, our friend Mark at Drobo said, I can give you 3. Who do you think should get them? And I thought, I bet Nels would like some. Cool. Yeah. Nels, do you have a pick of the week?
I do. Thank you for asking. So I thought, with our theme today, Nicole's really interesting and thought-provoking research that, and we spoke about this, this idea that microbes have really had a big evolutionary hand in possibly some of the major evolutionary transitions or breakthroughs. And so I kind of wanted to highlight on this point, or sort of amplify on this point, how we're increasingly recognizing the intimate and sort of important and complex role of microbes to our own development, evolution, and species. And so I've picked my pick of the week is this new book from Ed Yong, the great science writer who's come up a few times on our show. He just released this kind of much anticipated book called I Contain Multitudes, The Microbes Within Us in a Grander View of Life. And I think it's getting great reviews. And he really did both a careful job of kind of not overselling, but trying to really update the science in a way that's accessible, which I think is a really great goal. And I think he pulled it off without sort of overselling some of the hype of what the microbiome is and how we might manipulate it and so forth. We're kind of at the infancy of understanding a lot of these complex relationships, as we heard earlier today from Nicole.
Yep.
And so I've got a link here to, I just listened to this, I was driving this last weekend and listened to the Fresh Air interview with Terry Gross that Ed Yong did. It's a fun way to kind of get a behind the scenes look or insight into kind of how he thinks about the book and put it together directly from the author. And so I think it's maybe a half hour or so. It's a good listen.
Yep. Good stuff. Good book too.
Yep. Do you have a copy yet, or have you picked it up? Or
I have not. Someone picked it over on TWiEVO
I see.
when it first came out, and we talked about it. And Ed actually tweeted, "Thanks for the TWiEVO mention."
We'll see if he's tuning all the way into TWiEVO.
Yeah, I often feel that science writers don't really listen to podcasts. They have enough to do, and
sure.
I have a lot to read, I suppose. They want to formulate their own opinions. Although I think it would be a good idea if they did, listen to a couple of scientists talking about a paper might help. But they need quotes from people, I think, and that doesn't help them too much. So, but I'm surprised that he picked that up.
Yeah, pretty good.
He's a great writer.
Great writer. How about you, Vincent? What's your pick of the week?
Well, I am very lucky to be a volunteer at ASM, the American Society for Microbiology, and I get copies of books that are published by ASM Press. And I have one here that I got, which I love. It's called Infections of Leisure. It's a compilation of stories. The editor is David Schlossberg. But I just want to tell you the chapter names. I've looked at a couple of these, and they're really good. So these are infections you get under different situations. For example, infections from the ocean, infections from hot tubs, the camper's infections, diseases acquired in the garden from dogs, cats, birds, house pets, rats, petting zoos.
I love it.
This is a good one. Sexually transmitted diseases from boudoir to bordello.
Whoa.
Body piercing and tattoo infections. Infectious diseases at high altitude. Here's one for Utah.
There you go.
Infectious risk of air travel, cruise ships, and the last chapter, exotic cuisine.
Oh, yeah.
How cool is that? A book with all this stuff, which when you finish reading it, you're going to stay inside and never eat out, never travel, never go anywhere.
Yeah, never keep pets. I have sometime I'll have to tell my story. So I keep fish tanks, and in graduate school, I also actually got a Mycobacterium tuberculosis infection, fish tank tuberculosis.
Yeah,
but
that happened.
It's a longer story. We'll save that one for another.
That's one of these chapters, I'm sure.
Yeah, there you go.
This last chapter on exotic cuisine, is it, quote, Cantonese saying, if it has 4 legs and it's not a table, eat it.
Yeah, I'm guessing oysters might make a appearance there. Other raw.
Oh, yeah. Yeah, that would be in the ocean, yeah.
Uh-huh.
So if you want to know what you're in for by living on Earth, check it out. All right, let's go back to Justin's pick. Why don't you tell us what, remember Justin, he had a pick of the week.
Yeah, exactly. Let me just scroll up here. So here's Justin again. I also have a listener pick. I just happened to be reading Endless Form's Most Beautiful, The New Science of Evo Devo in the Making of the Animal Kingdom by Sean Carroll, who showed up earlier in our conversation where Nicole was a postdoc. On the same day, I was listening to TWiEVO number 6, the episode on butterflies. It is a fantastic book and is very fun to read, especially if you really want to think about the nuts and bolts of how evolution works in animals. I also thought it really complemented the discussion in your podcast. Yeah, thanks, Justin. I think that is a good connection to make. Sean Carroll is a great writer as well. We can go to our triple book picks of the week. Not only Endless Form's Most Beautiful, I think his new book, The Serengeti Rules. I haven't read it yet, but I got it. Someone gave me a copy. I think my dad passed it along to me, and I'm excited to dig into that one too. A lot of really cool stories, ideas, and Sean Carroll is another one of these great writers, scientists and writers.
This one sounds interesting. I'd like to pick this up.
Mm-hmm.
Sean Carroll is the head of communication at Howard Hughes, right?
Yeah, that's right. So he still has his lab running in Madison. I have a buddy who is in his lab working still, and they're doing some interesting projects on the evolution of snake venom, for example.
Cool.
But he's also splitting his time, I think, and in fact, spending a significant investment in science communication through the Howard Hughes in that position. So, yep.
I once wrote to him and asked if Hughes would consider supporting our podcasts. And of course he said no. So every time I see his name, I get a little ticked off, you know.
Maybe.
He said, we want to do our own thing.
Yeah, fair enough. But maybe we'll have to revisit this now that we've been, plugging some of his work, interviewing his trainees.
So one of the things they did at the Hughes, which is cool, is they started a movie production company called Tangled Bank Studios.
Yeah. And
recently they released a movie about zoonotic viral infections, which played on PBS. It was quite good.
Mm-hmm. And they also have, sorry, go ahead.
They have a publicity person there at Tangled Bank, and she wrote me and she said, could you give us some publicity for this movie? I just thought it was so funny, right? Because they turned us down, but now they want, because we're so successful, they want us to promote them. So I said, yeah, I'm happy to promote good stuff. Do you think you could give us a little love? Because you have so many, you have a wide reach. Just mention us, tweet us, and she said, yeah, she would do that. Although I haven't seen anything yet, but a mention from their Twitter would do a lot because they probably have lots and lots and lots of followers. So,
no question.
Nels, I try and do anything. I just, people say no, but I just keep trying. I figure slowly but surely.
Yeah. Well, it's just like these host microbe interactions, right?
That's right.
Keep colliding, and maybe a symbiosis will unfold somehow at some point.
I'm very patient.
I think, you and me both. I think Tangled Bank also has the rights actually to Ed Young's book. I think they're going to try to put together a
cool
movie based on that as well. So lots of connections there. Yeah.
Nice. All right. TWiEVO is at itunesmicrobe.tv/TWiEVO. And by the way, there are many ways to listen to TWiEVO. You go to iTunes, of course, but you can also use a podcatcher, which is a little app you can get for your iOS device or your Android device. Even if you don't use iTunes, I'd like you to go over there to the TWiEVO page and just rate the show. Of course, it's a 5-star show. We all know that. But just go over and rate it, and that makes it more visible in iTunes. When new people come and they look for podcasts in science, they'll see us. And I want to get more people to listen because we think this evolution stuff we're doing is really cool. We want more people to listen. So if you could do that for us, that would be great. Take you about 2 minutes. Also consider supporting the science shows of Microbe TV. You can go to microbe.tv/contribute. You can, give $1 a month at Patreon, or you can give us a little bit of money through a credit card or PayPal. just to help us do a little more with our shows. We can travel some more, do some remote shows, and so forth. That would help us out a lot. And, of course, always send your questions and comments to TWiEVO at microbe.tv. Nels Elde is at cellvolution.org. He's also on Twitter as Earlybird. Thank you, Nels.
Thank you, Vincent. Great as always. I can't wait for the next time in vivo.
And that was a good guest today. Really good guest.
Great
Nicole
guest.
King was terrific. She seems really smart and creative and actually seems like a nice person, too.
All of those things. Incredible scientists and incredible work. Yeah.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the sponsors of this show, CuriosityStream and Drobo. The music you hear on TWiEVO is performed by Trampled by Turtles. You can find their work at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.